Hello everyone. In the previous slide we have studied the monosaccharide and today we will study about the disaccharide and the polysaccharide. So first of all, what is the disaccharide? Disaccharide means when two monosaccharides they are joined together, then the disaccharide is formed. For example, here it is the maltose. The maltose is formed by two glucose molecule. That means one glucose molecule, when it is joined to the another glucose molecule, it is formed the maltose. So now after this maltose formation, there is a bond that is called as the glycosidic bond, or it is called as the O-glycosidic bond, because here in the middle of the bond there is a O, or the oxygen group is present. That's why it is called as the O-glycosidic bond. Now by this method a hydrogen molecule is released by this hydrogen and this OH. Now this bond is also called as the alpha 1,4 glycosidic bond. Why it is called as the 1,4? Because here the bond is formed by the first carbon and here it is the fourth carbon. So that's why it is called as the 1,4 glycosidic bond. Then why it is called as the alpha? Because here the first one, here it is called as the alpha. In the previous slide we have studied that what is the alpha. That means in the anomeric carbon only can be the first and the second carbon, and in the glucose it is the first carbon, and in the first carbon the OH group is present at the lower side. That's why it is called as the alpha carbon or the alpha glucose, and that's why the bond is called as the alpha 1,4 glycosidic bond, because alpha means here the OH group, because it is present at the lower side, it is called as alpha, and the bond is formed by the one or the first carbon and the fourth carbon of the glucose. That's why it is called as the alpha 1,4 O glycosidic bond, and the O glycosidic bond is called because of the presence of the oxygen in the middle of the bond. So that's why it is called as the disaccharide, and the maltose is formed. And the maltose, here the maltose is the beta maltose. Why? Because this is the anomeric carbon again, and in this the OH group is present at the upper side. That's why it is called as the beta. So it is called as the beta maltose, and the beta maltose is also called as the beer sugar. As the name suggests, this sugar, the maltose, is present in the beer. That's why it is called as the beer sugar. The second example is lactose. The lactose is formed by the galactose and the glucose, and the bond is called as the beta 1,4 glycosidic bond, because here the beta means here the oxygen is present at the upper side. That's why it is called as the beta, and 1,4 means the bond is formed by the first carbon and here it is the fourth carbon. So that's why it is called as the beta 1,4 glycosidic bond, and here also a hydrogen molecule is released. And beta lactose is also called as the milk sugar, because the lactose is present in the milk. The third example is the sucrose. The sucrose is formed by one glucose molecule and another is the one fructose molecule. In the fructose molecule the anomeric carbon is the second, because the first carbon here it is the first carbon and the second is the this one is the second. So always in the fructose the anomeric carbon is the second, and here the bond is called as the alpha 1 beta 2 linkage, because here this one is the alpha, and that's why it is called the alpha 1 because of the presence of at the first carbon. That's why it is called the alpha 1, and the beta 2 means here the OH group is present at the upper side. That's why it is called as the beta 2 linkage or alpha 1 beta 2 glycosidic bond. But here this is the different, because in the in the maltose here it is the anomeric carbon is left. That means any molecule can bind again to this group, and in the lactose also again the anomeric carbon is found. That means here also any molecule can bind, but in that case there is no alpha anomeric carbon is found, because one anomeric carbon at the second carbon and this one is the first carbon, they are already joined and there is no scope of the addition of the any molecule in this group. So that's why sucrose has no anomeric carbon, that's why it cannot add any molecule to this anomeric carbon. So again the sucrose is called as also the table sugar. Why it is called as the table sugar? Because earlier it was very expensive, so only rich people, especially in the western countries, they were able to afford this luxury. So that's why they started putting sugars on the dining table during their meals to symbolize that they are the wealthy and the rich. So that's why the term the table sugar was coined at the earlier stage. Nowadays, of course, everyone has the table sugar, so there is no luxury in nowadays. So the sucrose is also called as the inbred sugar. It is also called as the cane sugar because of the presence in the cane or the sugar cane. So again another is a trehalose. Trehalose is also joined by one glucose and another glucose, but here it is bond is the alpha 1. But here in the maltose the bond is alpha 1,4 bond. Why it is called the 1,1? Because here one glucose to this, the first carbon or the first hydroxyl group of the first carbon attached to this one at the first carbon, that's why it is called as the alpha 1. So in the trehalose it is found in the circulatory system in the insect. Another is the cellobiose. It is also a cellulose dimer and it is joined by one glucose and the one glucose, but here it is the beta 1,4. That means here the exception is all are the same, but here the OH group should be present at the upper side. That means at the upper side here it is OH and the lower side it is the H. Then it is called as the beta 1,4 glycosidic bond. Okay. So now reducing and non-reducing sugar. So what is the reducing sugar? Reducing sugar means any sugar that can reduce any molecule. That means, for example, this is the glucose molecule, they reduce 2CuO. That means 2Cu, I mean Cu2O2, and it reduces to the cuprose. This is the cupric, this is the cuprose. Why? How? Here this oxygen is joined to this group or the aldehyde group, and now it becomes the COOH, then it becomes the Cu2O or the cuprose. So that's why it reduces by oxidizing itself. So any sugar that can reduce any molecule is called as the reducing sugar, and this test is also called as the Felling's test. You must have done in the plus 2 exam. And second one is the again the picric acid. In the picric acid, this NO2 group is reduced again to form this NH2, and the whole group is called as the picramic acid, and it is called as the picric acid test. So here also this aldehyde group is oxidized to form the COOH. So that's why any sugar that can reduce any molecule like the picric acid or the cuprose or the ferric, and to form the, let's say this is the cupric, then after reducing it forms the cuprose. If it is ferric, then after reducing it forms the cuprose. If it is the picric acid, after reducing it forms the picric acid. So that's why it is called as the reducing sugar. Now come to the again to the previous slide. I have told earlier that in the sucrose there is no anomeric carbon is found. That means no molecule can be added to this molecule. So that means sucrose cannot accept any molecule, but the lactose, maltose, all other carbohydrates, they can accept the molecules or they can reduce the molecules. So like that, only sucrose and the trehalose are the non-reducing sugar. That means they both cannot reduce any molecule. All other biomolecules, means all other carbohydrates, they can reduce the any molecules. That means most of the all the sugars are the reducing sugar except sugar and the trehalose. Now come to the polysaccharide. As the name suggests, poly means many. That means more than two monosaccharides, when they join together, then it forms the polysaccharide. And the polysaccharide, again, it can be divided into homopolysaccharide and the heteropolysaccharide. What is the homopolysaccharide? Homo means same. That means here all the residue are the same. That is called as the homopolysaccharide. Hetero means they have also more than two monosaccharides, but they are different. They are not same. So first come to the homopolysaccharide. One of the most important example is the starch. Starch has the two component. One is called as the amylose, another is called as the amylopectin. So what is the amylose? The amylose means it is a linear strand of the glucose molecules. That means here it is a glucose molecules, here another glucose molecule, here another glucose molecule, here another glucose molecule, and they are joined by the alpha 1,4 glycosidic bond. And it has another component, it is called the amylopectin. In the amylopectin, it has same structure. That means one glucose, another glucose, another glucose, another glucose, and they are joined by the alpha 1,4 glycosidic bond. But at the interval of 25 to 30 residue, it is joined again by to the same structure, but here the bond is alpha 1,6 glycosidic bond. So that's why there are two components. One is the amylose. Amylose is the simplest structure, and the amylopectin, it is a branch structure or the complex structure. In the starch, both amylose amylopectin is present. Amylose are maximum 20 to 30% and 70 to 80% as amylopectins. So please note that a iodine test is used to determine the starch. That means if in the solution the starch is present, it reacts with the iodine and changes the color. For example, this amylose, the amylose structure is most responsible for the iodine test, because iodine can bind to this amylose structure, because this is a simplest structure and iodine can join together, can bind together, and it changes the color. For example, this is the starch. When the iodine is added, the iodine reacts with the amylose and it changes the color to the blue or the purple color. But iodine cannot bind so much to the amylopectin, because amylopectin is a complex structure. So that's why very low amount of the amylopectin bind to the iodine. So maximum responsible for the iodine test is the amylose of the starch. And then the starch is the storage form of the glucose in the plants. That means the one glucose, another glucose, another glucose, they are the store structure in the plants. Now come to the glycogen. So what is the glycogen? Glycogen is the storage form of the glucose in the animal. Like in the plant, the storage form of the glucose is the starch. So like that, in the animal, the storage form of the glucose is called as the glycogen. Here in the glycogen, again the same structure, amylose, amylopectin, they are present. But here the difference is that at the every 8 to 10 interval, the alpha 1,6 glycosidic bond is found, whereas in the starch about the at 25 to 30 interval the alpha 1,6 glycosidic bond is found. So the glycogen is similar to the starch, but it is very complex because of the presence of this alpha 1,6 glycosidic bond at the every 8 to 10 interval. That's why it is more complex. So glycogen is present at the liver and the muscle. So that means when there is low of the glucose in the cell, then these glucose molecules, they are set up from the glycogen and they mix to the blood, and here the process is called as the glycogenolysis. So next is the cellulose. So what is the cellulose? Cellulose is the linear on-branch structure, and the cellulose is the most abundant biomolecules in the earth. That means the carbohydrate is the most abundant biomolecules in the earth, and out of all the carbohydrates, the cellulose is the most abundant biomolecules. So it is the structural polysaccharide. That means it forms the cellulose fibers and it provides the rigidity to the plants. So it is simply a linear found by the beta 1,4 glycosidic bond, but the cellulose cannot be hydrolyzed or cannot be digested by human beings, because for the digestion of the cellulose an enzyme is required that is called as the cellulase, which is absent in the human. That's why if we eat any fruit or any grass, then only the cellulose of that fruit or the grass, they cannot be digested by the human being because of the absence of the cellulose. So now come to the next slide. This is called as the chitin. It is also again a homopolymer. So it is linear and it is formed by the beta by N-glucosamine. We have studied earlier that means in the glucose, here it is the OH bond is OH group is found, and in the glucosamine the OH group is replaced by, this is called the acetyl group COCH3. This is called as the NH2 or the NH group. This is the amine group, and by combining this is called as the N-acetyl glucosamine, or simply called as the NAG. So here all the NAG, the N-acetyl glucosamines, are joined together by beta 1,4 glycosidic bond, and the chitin is the second most abundant carbohydrate after cellulose. Cellulose is the most abundant, chitin is the second most abundant carbohydrate after cellulose. So the chitin is present in the cell wall of the fungi. It is also present in the exoskeleton of the insect and the crustaceans. So next come to the heteropolysaccharide. Heteropolysaccharide means here the polysaccharides, that means the residue are the not same. For example, the most important example is the glycosaminoglycan, called as the GAG. So what is the glycosaminoglycans? They are joined to, that means one is the acidic sugar, another is the amino sugar, then again acidic sugar, then amino sugar, then acidic sugar, then the amino sugar. So like that they are a stretch of structure that is called as the glycosaminoglycan. So what is the acidic sugar? Always the acidic sugar should be uronic. So what is the uronic? We have studied earlier in the slide. So that in the first slide, that when the, let's say it is the glucose, here in the terminal part is oxidized, then it is called the uronic. Why it is called the glucuronic? Because the monosaccharide is the glucose. That's why it is called the glucuronic. So uronic is the suffix. That means if the monosaccharide is galactose, then it is called the galacturonic acid. So always the acidic sugar is the uronic acid, and the second one is the amino sugar, and it should be the N-acetyl glucosamine or N-acetyl galactosamine. Okay, so there are the different types of the glycosaminoglycans. For example, the heparin. Heparin is the glucuronic acid with N-acetyl glucosamine. This is the first structure. This is the heparin. This is the second structure. That means ioduronic with glucosamine. So like that in the keratin, chondroitin, dermal sulfate and the hyaluronic acid. Hyaluronic acid, it is formed by the glucuronic acid with glucosamine, or simply called as the N-acetyl glucosamine. Okay. So always the glycosaminoglycans, they are the negatively charged, and the unbranched composed of the repeating disaccharide of acidic sugar and the amino sugar. So the glycosaminoglycan, they are found only in the animals and the bacteria. That means it is absent in the plants. So what is the function of the glycosaminoglycans? So they are covalently attached to the protein to form the proteoglycans. So here, what is the proteoglycan and the glycoprotein? That means any carbohydrate that can joins to the protein, it's called as the glycoprotein. But if the carbohydrate is the glycosaminoglycan, then only it is called as the proteoglycan. For example, if the glucose is bound to protein, then it is called as the glycoprotein. It is not called as the proteoglycan. The proteoglycan is called only when the carbohydrate is glycosaminoglycan. For example, the heparin. If heparin binds to the protein, then only it is called as the proteoglycan. But it has an exception, that is called as the hyaluronic acid. Hyaluronic acid cannot join, or they never join to the protein. So all the glycosaminoglycan, they can be the proteoglycan, or they can bind to the protein except the hyaluronic acid. Okay. So next one is the peptidoglycan. So this is also a heteropolysaccharide, or it is also called as the murine, and it is present in the eubacterial cell wall. Note that only in the eubacterial cell wall. In the bacteria there is a archibacteria, another is the eubacteria. So the peptidoglycan and the murine is found in the eubacterial cell wall. In the archibacteria, it is not peptidoglycan, that's why it is called as pseudomurine. You will study in the bacteria. So now this peptidoglycan is found in the eubacterial, and it is found by a NAM and the NAG group. What is NAM? This is the N-acetylmuramic acid. Again, it's a amino sugar, and NAG means N-acetylglucosamine. So one NAM, another NAG, they are joined together to form the peptidoglycan with the beta 1,4 glycosidic bond. So always 4 amino acids, they are joined together, and these amino acids are alternative. That means 1L, 1D, 1L, 1D. The first 4 will always alternative. So it is called as the tetrapeptide. Here it is called as the pentapeptide because of the presence of the 5 amino acids. But first 4 should be alternative one. That means LD, LD, like that. Okay. So that's why this is called as the peptidoglycan or the murine. So these are all about the carbohydrate. And if you like the video, then please like and subscribe our channel. Thank you.